Hi all, in session today we're going to be looking at the sensory system. In the last 2 weeks we would have started to look at the nervous system. We spoke about action potentials, transport, how they are transported across neurons, and then we looked at different structures in the brain, which leads us to talking about our five senses and the important organs of the sensory system and how they work. So what are sensory organs? These are receptors in our body that help to monitor the internal and external environments, as well as transmit signals from our peripheral nervous system to our central nervous system for processing. So this helps to connect those two major arms of our nervous system, your peripheral and central nervous system. The function of our sensory organs is extremely critical for homeostasis, for example, in thermoregulation and so on. It is because of those sensory receptors scattered throughout our body, they allow us to identify where there are spikes or drops in temperature and when our body needs to allow it to be returned to a normal environment via homeostasis. There are different types of sensory receptors. The functional types are based on the modality, that is the type of environmental change that they may sense. So for example, we have chemoreceptors. Those would be receptors that respond to changes in chemical concentrations. Example, responses to blood carbon dioxide levels being too high, causing reflex actions in our muscles. Olfactory, meaning our sense of smell, as well as our sense of taste. All of those are controlled by chemoreceptors or chemicals. They respond to chemical changes in the body or in the environment. Then we have our thermoreceptors. Those are receptors which respond to temperature changes. Those are present in our skin. Mechanoreceptors, which respond to mechanical energy. Example, those that are associated with touch, pressure, vibrations, balance, and hearing in the inner ear. Photoreceptors, which respond to light, controlling our vision. Nociceptors, which respond to tissue damages or pain. And then we have proprioceptors, which are those receptors located in our muscles, tendons, and ligaments, which would respond to how or allow us to move. So throughout our entire body, we have responses taking place because of sensory adaptation. There are two types of responses of the sensors that are in constant stimulation. So you can have either phasic receptors or tonic receptors. Your phasic receptors, they adapt to the environment, whereas your tonic receptors do not, right? So based on the type of action potential being generated, you can have your phasic receptors adapting to those to tell you whether you need to respond quickly or slowly, compared to your tonic receptors, which just give the same types of response generally all the time. They are slow in adapting to those changes. Generally speaking, when we have senses or the way we react to different things or our sensation, it occurs in a four-step process. First of all, you must have stimulation, where there is application of the stimulus. The sensors that are most sensitive to that stimulus are going to be activated. So if it is light, for example, we are responding to, we are going to have our eyes being the most sensitive there to that stimulus, so it will receive the stimulus. If we have something in the environment that has to do with pain, we're going to be activating our pain receptors and so on, right? The second step of sensation is transduction. When your sensor has been stimulated, it is going to now activate an action potential once it is strong enough. So if it is strong enough, it causes the depolarization of the nerve cells in that region, generating an action potential. And once the stimulus is above the threshold, your action potential fires a response. So it's going to trigger now that message to be sent up to your central nervous system through the process of conduction. So in your third step, because of that action potential being generated, high enough to generate, the stimulus is high enough to generate an action potential, it's now going to conduct that action potential to the central nervous system. So the information is relayed through a sensory pathway. And then in our central nervous system, we're going to have perception. So there's awareness of the environmental change. Your brain is aware now of something that is taking place in the environment. It's going to evaluate the nature of that stimulus and then send out a response. So those are your four steps. We had stimulation, transduction, conduction, and perception. Now, sensory inputs can be classified in a variety of different ways. We can have somastatic senses, where you have senses located over wide areas of your body. Example, your cutaneous receptors in your skin, your proprioceptors that activate your muscles. The type of information that is conducted here usually is generated up to your spinal cord first, and then it's sent to your brain. So those are the most common types of, or the most in number, the larger number of quantity there of sensory input that we would be dealing with. Then we can have special senses. So those are where you have specific sensory organs located in the head. They do not use the spinal cord as a means of transport because they are actually connected directly to the brain. So the information is conducted directly to the brain. Example there, our eye. It has an optic nerve that is attached directly to your eye that generates that information to that specific location of your brain. So it does not need the spinal cord or any type of other mechanism to transport the information. It's transported directly. You can also have proprioceptors, which I mentioned earlier. These are sensory receptors that are located in your muscles, tendons, and joint. They detect stretches of muscles, limb movements, position of body parts, etc. Example, you have your spindle fibers, your Golgi tendon organs. So this is an example here showing you your patellar reflex when you respond to a patella. This is a proprioceptor. If you all know, when you're testing your reflexes, they would use a patella hammer to shrug the ligament. That will therefore coordinate a response because it activates the sensory neurons that are connected there. This will activate alpha motor neurons that is going to then cause your knee to trigger upwards or give you that movement there for your patellar reflex. And then you can have cutaneous receptors. These are sensory receptors present in the skin, and they would respond to things like touch and pressure, heat and cold, pain. So you see there, they're naming a couple of them. You have your Mesenteric corpuscles, your Merkel discs, you have your Pacinian corpuscles. So we're going to talk about what those are specifically responsible for with regard to sensory or somatic senses. So how do we respond to different types of things on our skin? As we would remember, when we did integumentary system, we named a lot of different types of sensors that we have present in the dermis of our skin and how they respond to different things. So this might be a bit of revision from what we did early on in the semester with regards to integumentary system. We have heat, cold, and pain, the receptors that are responsible for that, and your free dendritic endings of the sensory neurons, right? So you'll see on your right-hand side, it's pointing to one of those free nerve endings. Those will detect changes in heat, cold, as well as respond to pain. Touch, you have dendrites that are wrapped around hair follicles that allow us to respond to touch. So sometimes things come really, really close to your skin. They don't actually touch the epidermal layers of your skin, but they touch your hair follicles and they respond to that touch, although you're not coming into direct contact with it. Your hair follicles have been activated because of the fact that there are dendrites wrapped around those, right? And those are referred to as Ruffini endings or Merkel discs. So you'll see them identified there in your diagram. Those are responsible for response to touch. And then you have response to pressure, where you have encapsulated endings, which are called Mesenteric or Pacinian corpuscles. Again, they are highlighted in your diagram there. And those are all located mostly in the dermal layer of your skin. So coming to our 5 different senses, as we would know, all of our 5 senses, they act as special senses because they are specialized to that specific function. So we have special senses that allow us to taste, smell, obtain equilibrium or balance, hear, and see. So we first talk about taste and smell. They are both activated by chemicals in the environment. So they are both made up of chemoreceptors. Taste, or taste cells, those are specialized epithelial cells that are found in barrel-shaped buds on the tongue. So you can see that barrel-shaped taste bud there, identified in your diagram to the right. They have microvilli which extend into the external environment. So you're seeing here your microvilli extending out of the bud as taste hears. They are not neurons, but they respond to the stimuli by also generating depolarization, similar to how our neurons act. And they would release chemical transmitters that stimulate sensory neurons. So you're seeing those yellow fibers at the bottom there, the sensory nerve fibers. And those are what are responsible for releasing neurotransmitters to allow us to detect different tastes, right? And you all would remember last week we discussed different regions of the brain. So they're asking here which cranial nerves carry taste information to the brain. So you have 5 different modalities of taste, or 5 different ways in which you can taste things. These are salty, sour, sweet, bitter, and umami. Umami is actually a type of a meat flavor due to the glutamate present in meats. Each taste bud contains cells that are sensitive to all 5 of these tastes. Previously, you will see in textbooks earlier on, they would have referred to different regions of your tongue that are concentrated with different types of specific taste responses like salty, sour, and sweet. That has been disproven, where we have found that taste buds actually all contain cells that are sensitive to the 5 tastes. It's just that some regions may be more sensitive to one type of a taste than another, right? A given sensory neuron is devoted to a single taste. So in your taste buds, you will have sensory neurons. You will have about more than, obviously you're going to have more than 5 types of neurons, or more than 5 neurons in numbers. One is specified to a specific taste. So let's say you have about 50 sensory neurons located in that taste bud. You can have 10 that are devoted to salty, 10 to sour, 10 to sweet, and so on. So how are particular taste detected? Taste and smell, I remember we mentioned previously, they depend on chemoreceptors, so they actually respond due to a chemical being present, right? So salty and sour taste, they are activated by passing through ion channels on the membranes of these cells in our taste buds. When we have sodium ions in a large present in our food, that's how we detect salty taste compared to when we have the presence of hydrogen ions in large concentration in our food, we detect sour taste. The other 3 types of taste, sweet, bitter, and umami, these are produced through the binding of specific molecules to protein receptors that are embedded in the plasma membranes of those cells in your tongue. Then you also have your sense of smell, which is also dependent on it's a chemoreceptor, so it's dependent on the presence of chemicals in the environment to be activated. Smell is also referred to as olfaction. So when you have chemicals that are present in the air, you're going to have action potentials generated due to those chemicals when they attach to the sensory cells of your nose, right? The specific sensory cells that are located in your nose. And you're seeing on the top right-hand corner there, it's labeled the olfactory bulb. That is the region of your nose where you have a concentration of those sensory receptors that's going to detect different scents in your environment. So when chemicals are present in the air transporting scents, they would bind to those sensory receptors in the olfactory bulb and generate an action potential, which will travel via an axon up into your olfactory bulb, right? So it connects there into that blue structure they call the olfactory bulb. In the olfactory bulb, the axon then synapses or connects to other neurons that are grouped in a glomeruli. So it's a glomeruli is referred to as a knot of tissues, right? So you have a knot of neurons there. So the olfactory bulb is going to transport that message there with that stimulus to all of the neighboring neurons that are connected or associated in that glomeruli. Each glomerulus will receive input from one particular type of olfactory receptor, and then this information is sent to your brain to be detected, or for your brain to decide what is the scent you are smelling. So whether it's a perfume, something that you know you can identify, which you have smelled before, for example, someone's perfume. So you know they're coming, the scent of gas, so you know that there's a leak somewhere, the scent of a specific food that you can identify because you have been exposed to it before, and so on. So something to note here, it is also connected to your limbic system. As you would know, different scents cause triggers of different types of emotions. So food can bring us smells of familiar foods that you enjoy can actually bring you sense of joy. Sense of perfumes that remind you of someone from your past that you may associate emotions with may bring you senses of joy or sadness, and so on. So you also have your limbic system here playing an important role in different scents. Then we have hearing. Hearing occurs because there's neural perception of vibrations or sound waves that are present in the air. We refer to different types of, the way in which we hear things depends, on the pitch of the sound wave or the frequency of sound waves. Humans can hear frequencies between 20 and 2000 hertz. We know that there are some animals that can detect noises below those frequencies, which we cannot hear. And we also hear things depending on its loudness, right? So this depends on the amplitude or the size of the sound waves, which is measured in decibels. Each 10 decibel increase represents a tenfold increase in amplitude or the size of the sound wave. So when we hear things, it depends, we discern what type of sounds we hear based on their pitch and their loudness. Our ear is the organ that's responsible for our sense of hearing, right? Some of the major structures, we have our outer ear, our inner ear, and our middle ear. So we're going to talk about the different structures found in those different parts of our ear. So we have our outer or external ear. It's made up of our pinna, which is what we actually see as our ears externally, right? So it's that extension of your cartilage, just for funnel-shaped kind of structure. It's called also the auricle. It's responsible for connecting and channeling sound waves. Then we have our external auditory meatus, which helps with conduction of sound waves. So that's our little canal that leads into our ear. And then we have our tympanic membrane, which is located under that, or which is located around that tunnel, I should say. It vibrates when it's struck by sound waves. So if you look at your diagram there, the helix, the auricle, the earlobe, all of those things make up your pinna, and then it funnels that sound or those sound waves into the external auditory meatus, which will then be transported into your middle ear, right? Your middle ear is made up of, so you'll see in your diagram there above, it's showing you the region of your middle ear. It is generally an air-filled chamber which contains your auditory tube or your eustachian tube, which connects your middle ear to your pharynx, right? There are three major auditory ossicles which act as sound amplifiers. These are like three little bones. So you're seeing that blue structure on your diagram on your right. In the region of the middle ear, you all are seeing those three little loops. There are two extending upward and one extending sideways. Those are our three auditory ossicles. You have your malleus, you have your incus, and your stapes. So they would act as amplifiers, right? So you're seeing here, then it leads into, into your inner ear. Your inner ear houses the structures for senses of equilibrium as well as hearing. So it consists of a bony labyrinth and a membranous labyrinth. In the bony labyrinth region, you have a series of curved passages within your skull bones. There are two distinct regions, your cochlea, which is responsible for hearing. So you're seeing that little shape there that's looking like kind of like a snail shell. That's your cochlea, which is responsible for hearing. And then you have your vestibular apparatus, which is in your diagram highlighted or labeled as a yellow structure, right? And that's responsible for equilibrium. Within there, you have several semicircular canals which assist in how we maintain balance. So we'll talk about that in a bit. Then you have your membranous labyrinth, which is a membranous network that parallels your bony network, and it contains fluids like your perilymph and endolymph, all of which play a role in how we hear and how we maintain our balance. We're not going to go into detailed information on how those work. So what's your cochlea? Your cochlea is very important. It is responsible for how we hear things. It's the part or specific region of your ear that is responsible for generating sound, right? So it's located in the anterior portion of the inner ear. You also have hair cells that are present there that change your sound waves into action potential. So it is in this region that the sound waves are going to now generate an action potential in those specific nerves or neurons that are responsible for hearing. You have three snail-shaped tubes that are filled with fluids, right? They are your scala vestibuli, your scala tympani, and your cochlear duct. So you're seeing there, if you look at your cochlea, your little blue diagram, again, you're seeing three different regions. Those are the regions that would line up with your scala vestibuli, your scala tympani, and your cochlear duct. And those contain different fluids that are going to help in the transport of your sound waves and the conversion into an action potential. In your ear, in the next region of our ear, which is, we looked at it earlier on your vestibular nerve, in that yellow structure there that is highlighted in your inner ear, those are responsible for your sense of equilibrium or how we balance. So you have mechanoreceptor cells that are also responsible for equilibrium. They possess hair-like extensions known as stereocilia on the surfaces that are highlighted in blue in your diagram, and along a large kinocilium, which is in pink in your diagram there. So let's just discuss briefly what happens with the movement of those hair cells that allow us to maintain balance. The bending of your stereocilia alters your membrane potential. It causes a depolarization, which then induces more neurotransmitters to be released to the associated sensory neurons. So in diagram 8, it's showing you an electromyograph of what those look like. At rest, you are seeing your kinocilium and your stereocilia are both standing upright at rest. When they are stimulated, they move slightly to the left. So when an action potential is generated, it causes the membrane to be depressed. The plasma membrane becomes depressed and causes those cilia to shift to your left. Whereas when it's inhibition taking place where your action potential is leaving, it then causes them to move slightly to your right, and then they would return to rest. Now, I forgot to mention, based on the movement of these structures, it's going to trigger a response that will cause us to maintain our balance. So when those cilia move to your left, for example, it's going to trigger a response by our muscles in different parts of our body to allow us to maintain balance. So let's just take, for example, if we've been spinning in circles for a while, when you tend to retain balance, when you're going to stop moving and you want to regain balance, your muscles in different, because of the movement of the hairs that was detected in motion, the muscles in your different parts of your body are going to respond accordingly to ensure that they would move according to which way will best allow for you to balance properly. And then you have your organ of Corti, which is your spiral organ located in your ear. It forms the floor of the cochlear duct, right? You have hairs that are embedded there, and they are responsible for stimulating your sensory neurons to produce action potentials, right? Again, we do not need to know detailed information on how these different specific organs in your ear work. We just need to be able to identify your cochlear and your vestibular nerves that assist in hearing and balance respectively, as well as their location. If you are asked to label them or identify them in the region of the inner ear, you should be able to do so. So, how do you have neural conduction and perception of auditory information or things or sounds in your environment occurring in your ear? The fluid pressure waves causes the basilar membrane to vibrate, right? They are louder sounds. If you have a louder sound in your environment, it causes bigger vibrations, causing a higher frequency of action potentials. If you have softer sounds, the opposite will happen. You'll have smaller vibrations and lower frequencies of action potentials. Sounds can occur based on the pitch. So you can have sounds of different frequencies or pitches. Low pitches vibrate membranes closer to the apex, whereas higher pitches vibrate membranes closer to the base. And this is how we differentiate according to different pitches or frequencies of sounds. The hair cells would move against the tectorial membrane, causing the stereocilia to bend, which will stimulate neurotransmitters to release sensory neurons, resulting in signals conducted to your auditory cortex or that region of your brain that is responsible for hearing. So your ear is another organ that does not use your spinal cord as a means to transport information. It does so directly because of its connection, the auditory cortex being located very near the region of your ear and the inner canal of your ear. It is transported directly to that region of the brain to be processed. In the region of your semicircular canals, where they are located, these are 3 fluid-filled circular tubes that are oriented on 3 different planes. They're giving an example here so that you can understand how they work. So you're seeing in image A, the gentleman is facing forward, whereas in image B, he's moving left to right, doing a rotation, right? If you turn your head to the right, the endolymph of the lateral semicircular canal causes the copula to bend to the left, so that message is sent that you are turning to the right. So the movement occurs in opposing directions, right? So a rotation of the head in one direction can generate initial pressures in your endolymph, which can cause your movement to occur accordingly. Right, so when you have your initial pressure in the endolymph being generated, it causes the pushing and the bending of the copula, just similar to the way in which a wind pushes a sail, which is going to push the hair cells in one direction that stimulates your vestibular neurons to tell your brain. So if your movement is detected to the left in the region of your copula, it's telling your brain that your head is moving to the right and vice versa. You also have your otolith organs that are present in the region of your ear. They are found within two fluid-filled chambers, and they would detect linear movement or tilting of the head, bent forward or upright. So you have your otolith organs responsible for detecting that type of movement. Then it leads us to vision, one of our last major sensory organs. How do we visualize things? Perception of different types of objects in our environment is done through electromagnetic radiation, right? Our eyes can actually see a very narrow portion of the EM spectrum. We have photoreceptors that are present in our eye, which is the organ responsible for sight. They are stimulated by photons of light and they contain photopigments. They would undergo chemical changes that respond to light, and they would induce metabolic changes in your photoreceptors that will lead to your receptor generating an action potential that allows you to visualize different objects. So just a bit on the anatomy of the eye. You're seeing on your diagram to the top there, the major regions or the major parts that make up the organ, the eye organ. We just discussed some of those major parts of the eye. The sclera is the part of your eye or the white of your eye. It's made up of tough connective tissue, and it helps to protect the inner structures as well as maintain the shape of your eye. Then you have your cornea. Your cornea is that region of your eye. It's transparent that lets light pass into the region of the eye. So if you look at the middle, if you're seeing my cursor here, your cornea, it surrounds the eye and it allows light to pass into your eye. It's made up of a fixed convex lens to help bend light inward so that it's directed into that organ, and it covers the anterior chamber, and it's filled with a substance called aqueous humor that assists in the protection of that organ. Then you have your iris. It's a thin ring of pigmented muscle that's located in the front of the lens. The pupil, which is located in the opening at the center, which is the opening at the center of the iris. And then you have so the muscles in the pupil help to alter the size of your eye depending on how much light is entering your eye. So you enter into a dark environment, it's going to cause the pupil size to increase drastically so that it allows as much light as possible in that dark room to enter your eye so that you can try to see what's going on in that dark surrounding. Compared to if you're in a region or in a room that has really bright light, you're going to have your pupil size becoming much smaller because of the amount of light that it's responding to, okay? So the radial muscles are responsible for opening the pupil to let more light in. They are responsible for the sympathetic response of your parasympathetic nervous system. Okay, so that happens when you're in a dark environment. And the circular muscles cause the closure of your pupil when you're in extremely bright light. And that's a parasympathetic response that would occur if you're in a bright environment. You have your lens. Those are solid but pliable, transparent bodies. They are used to focus light onto the retina. So you're seeing on your diagram to the right here, the lens. That's that creamy colored structure there. It helps to focus the light onto the region in the back here where your retina is present, so that's where you have your receptor cells that are responsible for how light is received, right? Then you have your ciliary bodies. These are ring-shaped smooth muscle structures that encircle the lens, and they allow the adjusting of the lens in shape to allow the light to be focused onto your retina. And you have your posterior chamber, which is the space between the iris and the lens or the ciliary body. So accommodation. Accommodation allows your lens to change shape to focus light from objects at different distances on the retina. So no matter how far, how close something is, the lens is going to change shape in order to ensure that those objects are focused onto your retina, where you have your receivers or your receptors that are going to convert that stimulus into something that you can visualize. How is this achieved? It is achieved by your ciliary body, which allows that lens to become larger or smaller, right? So for far objects, light from the object would enter your eye from a narrow range of angles, causing the ciliary bodies to relax, causing the lens to stretch so that it becomes less convex and there's less bending of light so that those images are focused onto the retina. And the opposite would happen for near objects. Light from the object would enter the eye from a wide range of angles because it's closer to you. The ciliary body will therefore contract, causing the lens to recoil, and the lens becomes more convex and there's more bending of light because the object is closer. It needs to be bent in more to be directed onto the retina. There are some refractive disorders that can affect how your retina, how images are transferred or conducted to your retina. So some people suffer from myopia or nearsightedness, where they have a slightly elongated eyeball. Some people suffer from hyperopia or farsightedness, where they have shortened eyeballs, or some people suffer from astigmatism, where their eyeball is actually oblong shaped, and you have differences in refraction, so things are not visualized. In all of these cases, depending on the deficiency or the disorder, things are not directed onto the retina as they should. Going back to a bit of anatomy, you also have your vitreous chamber, which is a large chamber posterior to the lens and ciliary body. Your retina, which I mentioned, it's located in the lining of the vitreous chamber. It contains your photoreceptors. So extremely important part of your eye here where we have our receptor cells. It's made up of the fovea centralis, which is the point where light from the center of the visual field is focused, and it has a high density of cone cells in this region. And you have your optic disc, which is the region where the optic nerve joins the eye. There are no photoreceptors present there, and it's referred to what we refer to as our blind spot. So what are those photoreceptors present in the retina cells? We have our two photoreceptors, which are our rods and cones, right? We have bipolar cells as well present in our retina. Those are the neurons that receive neurotransmitters from the photoreceptors, and they synapse with your ganglion cells, so they transport messages from they would transport messages from your photoreceptors to your ganglion cells. And the ganglion cells are the neurons that receive neurotransmitters. They help to conduct action potentials directly to the central nervous system there. So you have light is directed onto the photoreceptors, which are your rods and cones. It will then move to the bipolar cells, which will then send a message to the ganglion cells, which will transport it directly into your central nervous system so that you can determine or discern what you're seeing externally. So what are those photoreceptors? You have your rods. Rods are generally more numerous than cones. They cannot distinguish different colors. They distinguish black and white types of light, right? So many of these rods converge into ganglion cells, and they are highly sensitive to light. So they can detect light in very low levels of light. So in a dark room, for example, when you're seeing things that are looking all black and white because you cannot discern color, those would be when your rods are activated. And then you have your cones, which distinguishes among colors. There are few cone cells that converge on single ganglion cells, and they generally need more light to see. So you, as with color, you would only be able to discern color if you're in an environment that is highly illuminated, and the images are much sharper than those that are projected by your rods. Each photoreceptor has two major segments, inner and outer segments. We're not going to go into too much detail there. You all don't need to know inner structures of those photoreceptors. And then you also have photopigments. You have rhodopsin, which is a major photopigment present in rod cells, right? It consists of different types of vitamins that allows the generation of action potentials to allow you to see in darker environments. And then you have your cone cell photopigments. Each one has responses to different wavelengths of light. So it can respond to blue, green, or red lights to allow for discrimination of different colors in the environment. Okay, so that brings us kind of to the end of what we were talking about with regards to sensory systems. Just to recap some of the things we did, we looked at the different types of sensory receptors. We talked about what they do. We talked a bit about how sensation occurs, the 4 steps of sensation: stimulation, transduction, conduction, and perception. We looked at the different types of senses: somesthetic senses and special senses. In your somesthetic senses, you have your proprioceptors and your cutaneous receptors that allow for detection of different things. And then we went on to talk about our specific organs that are responsible for different things. So taste and smell with your chemoreceptors, your taste buds, versus your hearing and the different regions of your ear, right? We did that in a bit of detail, and then we went on to talk about the different parts of your eye and how they are responsible for how we see. So that brings us to the end of your lecture on sensory system. There's also a summary that you all can use that gives you a lot of good information on the different parts of your different organs that make up our sensory system. Okay, thank you.